Scott is also a former K12 scholar with the UCSF and Kaiser site, and he is giving a talk on defining and targeting aging mechanisms of urinary symptoms in older men, starting with mitochondria. And Scott is a faculty member at UCSF.
Thank you, Marvin. Really great to be here with you all in person. Please do reach out to me by email. I'm also on Twitter if you have any questions or want to discuss collaborations. I have no relevant disclosures for what I'm discussing. So I always like to start with what my research mission is, which is to help treat older adults with lower urinary tract symptoms, or LUTs as I'll refer to it for the rest of the talk, by studying how aging cells, tissues, organs, and systems contribute to these common urologic symptoms. And I'm an epidemiologist by training turned primary care doctor, so that informs a lot of the methods that I use. Here's a brief outline of what I'll go over today. And since I'm going to get cut off 15 minutes, I'm not going to go over it in detail. So this paper really highlights my method of my kind of whole research program. It's focused on age-related diseases and specifically this idea of health span, how many years you live without chronic diseases. But I've really used this as a model to see how epidemiology can inform the study and development of interventions to target age-related LUTs. So a couple of highlights, and these are all approaches that I have used personally and have kind of evolved from the top bullet point to the bottom throughout my career. I think frame my approach very well. So if you're interested in epidemiology, I recommend that paper. So this is mostly for the trainees in the room, but this is really my kind of anchor that drives what I do in the research questions that I ask. Specifically, my goal is to improve clinical outcomes. And so in the world of benign urology that I'm most interested in, LUTs, that includes bothersome LUTs. So if someone has LUTs, but they're not bothered, it's not a clinical outcome to me personally. If they develop acute urinary retention, have to go to the emergency room, they get hospitalized, things that require urgent intervention, obstructive nephropathy, kidney stones, recurrent UTIs, prostatitis, these are all clinical outcomes. There are 1 million age-related changes in the genitourinary tract, as well as the entire body, that do not meaningfully increase risk of clinical outcomes, whether or not that's age-related LUTs or other age-related diseases. And that is really, really important to take into account. If you look at older men, and older women, there are a bunch of things different about them than when they were younger, and they also have symptoms. That does not mean that those differences are causing those symptoms. I don't want to say anything inflammatory, but a couple of things that a lot of us use in our research such as detrusor instability, prostate volume, and urinary flow rate, if you look at the epidemiologic literature, are very weak predictors of clinical outcomes in humans. And that doesn't mean that they aren't worthwhile things to study, but we either need to measure them more accurately or figure out why it is that they are not explaining a large amount of variability in human disease and human symptoms, or we need new targets. And both of those are really worthwhile goals. But it's important to acknowledge that the amount of variability in symptoms that is explained by these factors is less than 10%, and in some cases less than 5%. So epidemiologic studies are critical for identifying modifiable risk factors that explain sufficient amount of variability in humans in these clinical outcomes that they are worth targeting in our gold standard levels of evidence for interventions. So the modifiable risk factor that I am most passionate about is age. And so older age is associated with greater LUTs attributed to BPH incidence as well as progression. If you're older and you don't have LUTs, you're at higher risk of developing it. If you're older and you already have LUTs, you're at higher risk of it progressing and getting worse over time. This is from the Health Professional Follow-up Study. And so most people have kind of ignored this. Age is, time moves forward. There's nothing you can do about it. But increasingly, there are several really innovative thinkers around what is it about chronological age that contributes to increased risk of many different diseases. And they've organized it into these buckets. There are the changes that occur at the cellular and molecular level called biological aging. These are also the hallmarks of aging that you might have heard of before. There are things that affect, that is called phenotypic aging, and as well as functional aging, which are more on the system end of things. And here I've listed all the things that I've seen individual papers saying that they identified a new mechanism of LUTs BPH in men. thinking of it in its silo. But if you take a step back, when you kind of put it all together, these are all manifestations of aging. And what is really exciting is that there's an entire new field called geroscience that is specifically looking at targeting mechanisms of biological aging, as well as its downstream consequences in phenotypes and functional aging. Caloric restriction and physical activity are kind of the quintessential early geroscience interventions, but there is a whole field developing pharmaceutical agents and targets that specifically target specific or multiple mechanisms of biological aging. And so this is how I'm kind of synthesizing all of the epidemiologic data that I've been generating, as well as others have, in order to develop better interventions. So I picked one of those specific biological mechanisms of aging, which is mitochondrial dysfunction, to kind of test this framework and really try to work out some of the kinks. One of the reasons why I chose mitochondria is because in the few studies that have looked at this, people can both develop mitochondrial dysfunction with age, or they can inherit mutations that cause mitochondrial dysfunction. And when you look at people who have these inherited mitochondrial diseases, and you compare them to age and gender matched controls, they have more LUTs, whether it's urinary urgency or weak urinary stream. And even more convincing is that there seems to be a dose response in terms of if you have the inherited mitochondrial disease, whether you have a relative that has one of the mutations, but not fully penetrant, versus controls, there seems to be a dose response. The more of these inherited mutations you have, the more severe your LUTs are. So I thought that was enough to kind of pursue this idea. To investigate this, I applied and got a O'Brien Center Pooled Opportunity Award. The way that mitochondria become dysfunctional in the body is extremely complex. There are entire conferences just focused on the single organelle. So I picked a couple of ones that I happen to have mentors or advisors who were experts in that pathway, and I started there. But that was the main point of that, was to show you how complex it is. I'm going to tell you about preliminary data from one of the aims, aim 2, because this is a session focused specifically on men. But we're also doing this concurrently in women. And my research question was, does mitochondrial content, protein expression, oxidative capacity, and bioenergetic profiles differ between BPH and non-BPH prostate tissues? And you can see that my hypothesis was that they would, and specifically they would be lower or more dysfunctional in the cases. To address this, I used homogenized prostate tissue from 89 prostatectomy BPH cases at UT Southwestern and 13 donor controls provided by CAIRIBU member, Dr. Doug Strand. I then helped get those tissue samples over to Dr. Anthony Molina at UCSD. He's an NIA-funded mitochondrial research expert, also leads their bioenergetics core. And to start, this is a part of kind of a multi-year program that we're working on. We started by measuring citrate synthase activity, which is one of the leading markers of intact and functional mitochondria, as well as their oxidative capacity. So here are my preliminary results that are some of you who went to the spring symposium have seen. We started off with those 13 controls. Again, the age of the donors, unfortunately, mostly die related to diseases that tend to be on the younger side, so such as accidents. But you can see, even just with these 13 cases, that there seems to be a potential slow decline in citrate synthase activity with increasing age among the donors. So what I expected was this to continue to go down, or to be even maybe disproportionately low relative to these donors. And so here's what we found for the BPH cases, which was the opposite of what I hypothesized. Sometimes it is fun/hard to say that you're wrong. But when we looked at the citrate synthase activity in the BPH cases, and again, these are fully homogenized prostates, we'll get to that a little bit later, very clearly the potential age-related decline that was observed in donors did not bear out. And given that these are all older individuals, the citrate synthase activity was clearly higher. And so we kind of tested that with comparison to means as well. So why might that be? At the spring symposium, I started to kind of hypothesize and get some advice from the group about what we should do next to investigate this. What we did first was we started with the BPH cases. And we said, okay, among those who have high citrate synthase activity and those who have the lowest citrate synthase activity, so extremes, what do their core, what is the protein expression from Western blots of the 5 complexes of the electron transport system look like? And what we found was that looking at BPH cases only, and so we haven't done the controls, which is going to be really important and interesting to look at, but among the cases, those with the highest and the lowest levels of citrate synthase activity had identical protein expression for all 5 complexes. The second thing, and it was somewhat blasphemous to even propose that I was going to homogenize the entire prostate and run these assays in front of this group that is very tuned in and has demonstrated the importance of heterogeneity in prostate, BPH prostates. So I got the feedback, are these predominantly glandular, predominantly stromal? We're just starting to investigate this. The easiest way, that was a suggestion of Dr. Strand, was he can visualize and just tell me like very crudely if it is predominantly glandular stromal, and even could put a number on it. We've only done it for about 15 of the samples, and I can't really tease apart a trend here. And so you're just gonna have to kind of wait and see for what we discover later. But the main conclusions, and the reason I'm presenting this to this group, is that showing you preliminary work, how we're progressing with our Pooled Opportunity Award. But our hypothesis was not only disproven, but it was in the opposite direction of what we expected. There is a lot of work to be done, and it's not exactly what I thought it was going to be at the beginning. Age-related decline in CSA activity was observed in a limited number of donor controls, and so that really motivated us to try to get more donor cases from Doug, particularly on the older age spectrum. And it is unknown if mitochondrial content is higher, which is what we hypothesize for why our hypothesis is wrong, or oxidative capacity of mitochondria is increased, which would be completely in the opposite direction of what we hypothesize, potentially due to increased energetic needs of hyperplastic prostate. What we're going to do next is try to tease out whether it is mitochondrial content, oxidative phosphorylation efficiency, whether there is some component of glandular versus stromal predominance that's driving this. And then we have other mitochondrial proteins that we are looking at next, as well as investigating things in the controls. The circles are some of the key proteins that we're going to look at. In the last 2 minutes, I think I have, and then we're repeating it in bladder tissue, which we're excited to look at specifically because I said that people with inherited mitochondrial disease have LUTs. I did not say that it was because of the prostate, and I actually have no idea which organ or which organs or systems are driving it. And so to narrowly focus on one could potentially miss what is truly driving the severity of these men's BPH. So because I said I'm an epidemiologist and I span, what I just showed you was a lot of tissue and running assays that I have no expertise in, but I'm developing a team and collaborators that do. I'll tell you a little bit more about my work that is kind of the predominant thing that the way that I'm kind of building out this framework. The first is the study of mobility, muscle mobility and aging. It's a prospective cohort of 900 older adults, all 70 and up. We added a LUTs questionnaire, the Learn SI10, at every annual visit for the entire cohort. And then this cohort is one of the most deeply phenotyped cohorts in existence, specifically with respect to age-related changes in muscle. And they do have entire mitochondrial team. They're getting thigh muscle biopsies as well as blood-based mitochondrial biomarkers. And so we'll be correlating that with changes in urinary symptoms over time. We also have full body MRIs, so we can also look at the urinary tract. And then I have some exciting news to share with this group, which is a CAIRIBU success story. So we applied for funding for a trial with Dr. Allison Huang, who is at UCSF with me. Dr. Kevin McVeary is also a co-investigator for a pilot RCT of 70 physically inactive men, aged 60 years or older with moderate to severe LUTs presumed to be due to PPH. We got a score under the pay line, but the funding decision is pending. The intervention is a multi-component aerobic and resistance exercise prescription with weekly coaching compared to health education control. Our outcomes are those clinical outcomes that I was referring to earlier: LUTs severity and bother, We will also be looking at urologic function and physical function. And then we're collecting a lot of biospecimens pre and post intervention. The reason I bring this up is that we will have a lot of biospecimens, a lot of data, and a lot of interesting opportunities that are not specifically what we were funded to look at. But if there are interested collaborators, I'd be more than happy to talk to you about how we can work together. And that's just what will happen to the people. And thank you to everybody for your attention, and to the organizers, especially the O'Brien Center.
Thank you, Scott.
All right, so we'll have questions held until the end. So I'll go ahead and introduce our next speaker, who is Dr. Petra Popovich, who is an assistant professor at Eastern Virginia Medical School.
I think I got it. Okay. Well, thank you very much for the invitation to present, and it's really good to see you in person finally. So I just recently became an assistant professor. So the work that I'm going to present is basically, what I started and I'm trying to continue. And I will also present some future aims. My work and my lab's work is focusing on prostatic inflammation. And when I talk about LUTs and BPH, I like to talk about it as a combination of different pathological processes. We know that proliferation, prostatic enlargement is really important, and smooth muscle dysfunction as well. But a lot of you already know that prostatic inflammation and fibrosis are really important key aspects of the disease. But the problem is that currently these are not therapeutically targeted. And we just don't know, I believe that we don't know enough about prostatic inflammation, and especially what inflammatory cells there are, what the immune landscape is. And I actually collected just a few studies from others that have been focused on what immune cells there are in the prostate in health and disease, just to have, like a better perspective. But this is not, if you feel like that I did not mention a study, it's not a full collection, but just some really interesting studies. So there is this study from Cambridge doing single cell RNA-seq. That's basically what we are now using to identify different cells. It's harder to identify immune cells because obviously, there are more epithelial and stromal cells, but what they found is that the majority of cells were of T cells and macrophages. And T cells is like the regular types that you would find. I'm always more interested in macrophages, and they found tissue resident profile macrophages as well as monocyte-derived but newly infiltrated cells. But what was really interesting in this study is that they found a macrophage subset, which was specialized for the high zinc content of the prostate. We know that the prostate has a really high zinc content, and these macrophages contain cysteine-rich proteins, metallothioneins that bind zinc. And why is it important? They depleted macrophages, and they found that the depletion of macrophages actually decreased zinc levels. And we know that zinc levels are actually correlated with prostate cancer and BPH as well. And another study, a lot of you are familiar with the Hayward group's findings. This is a study led by Renee Wickman, and they basically compared small and large prostates, and they enriched for immune cells with CD45, enriching for CD45 positive cells in small and large BPH tissues. And they also found that the majority of immune cells are T cells and macrophages. And they also found that a really important increase in TNF production in T cells and macrophages as well. And we are expecting some more results from these studies, these sequencing studies, to have more information about the different macrophage subsets fairly soon. And I also want to mention findings from the Tomi Cat Lab. They are really focused on Mastles, and it seems that mast cell numbers are increased in the hyperplasia in BPH, and the inhibition of mast cell activation alleviates urinary dysfunction in mouse models. So they are focusing on that area and that cell type. Our approach has been to look at a mouse model of BPH, which is produced by the implantation of testosterone estradiol pellets. And you can see that the highly enlarged bladder shows that there is a high obstruction, urinary dysfunction in this model. And we also know that the pathological changes that are found in human BPH are represented in this mouse model, but there are controversial information about the state of inflammation in this model. And so we looked into the inflammatory cells and inflammatory signals early on in this model, 2 weeks after hormone pellet implantation, and we did a nanostring screen. And the good thing about that screen is that it also provides you a cell type analysis. And what you can see here is that the most pronounced cell types were macrophages that were elevated in this model. And if we look at the top 3 genes, you can see SPP1, MMP12, serum amyloid A, but I just would like to focus on SPP1, which is otherwise known as osteopontin. And when we looked at the localization of these cells with in situ hybridization, we saw these cells in the actual prostate lumen that were highly upregulated, that had highly upregulated osteopontin expression. I had to redo the staining again because I couldn't believe that this was actually a real signal, because you can see that it's an aggregate. This is not how in situ hybridization looks like at all. When you look at it with fluorescent staining and fluorescent in situ hybridization, you can see that it co-localizes with the CD68 marker, which is a macrophage marker. So these are actually macrophages that infiltrate in the prostate lumen. And when you look at the actual, when you look at it with immunohistochemistry, you can see that I don't know if the cursor, oh, it does not work. So you can see that actually they have granular staining with CD68 and vimentin, and they occur both in mouse and human BPH tissues as well. And from their staining, what we thought that this granular staining was really interesting, and they might be actually foam cells. To identify foam cells, we have to use frozen sections and lipid staining. So we use OIRED O, and we could identify that there are actually lipid accumulation in these cells. So these are definitely foam cells. Foam cells, if you're not familiar with, they are lipid accumulating macrophages. And we don't know much about what their role would be in the prostate, especially not in the lumen. We also looked at whether these are M1 or M2 macrophages using iNOS and arginase 1, and they seem to have more arginase 1 staining, but it's at the same time there is iNOS expression to some extent. So this might be a dynamic macrophage subset that can go to one stage to another, and especially these are foam cells, so we don't know what they are and how they function. And because they accumulate lipids, we were interested in whether there is any lipid dysregulation in this model. And in the nanostring panel, there's only this one gene that is really connected to lipid synthesis, which is fatty acid synthase, and we found that it was significantly upregulated in the model. So there is probably a higher elevation in de novo lipid synthesis. And we also looked at human BPH specimens in terms of seeing the bigger picture of lipid accumulation. And this is just an N of 1, but it's representative of having a local upregulation potentially in larger nodules in this case. And that's really interesting, but we will follow up on doing an actual study of looking at the pattern of lipid accumulation in BPH. And why is it important to have luminal macrophages? What we think is that these cells can actually, whatever they secrete, it can reach more luminal cells, more epithelial cells in the prostate. And with the example of looking at secreted osteopontin with immunohistochemistry, you can see that the neighboring epithelial cells actually take up osteopontin. And so then we were interested to see the accumulation of immune cells in the tissue itself, both in T+C2 treated mice and in osteopontin knockout mice, to see what would happen if we remove osteopontin secretion in these cells. And you can see that the overall immune cell numbers are elevated in the model. So there is an increase in immune cell numbers. And mostly in the ventral prostate, what we see is that there is an increase in macrophages in the tissue. And that was also expected from the nanostring analysis. But when you look at the dorsal prostate, what's really important to see is instead of macrophages being really pronounced in this model, Mastles were elevated in response to testosterone estradiol supplementation, as well as this was inhibited by in osteopontin knockout mice. Also, we looked at urinary function by Voice-Pod assay, and you can see that there was a delay in the osteopontin knockout mice to develop urinary dysfunction. As well as we looked at fibrosis, and for fibrosis, you have to go out to 3 months in this model, and you can see that collagen as indicative of fibrosis was elevated in the wild type animals, but was reduced in osteopontin knockout mice. So altogether, this is not a full picture yet, but we could actually identify a mechanism of foam cells migrating into the lumen via steroid hormone implantation. And we know at least they secrete osteopontin, and they might have a direct connection with urinary dysfunction. And this is in the ventral prostate. If you look at the dorsal mast prostate, mast cells are potentially have a more important role. And at this point, it's really hard to determine why we have this ambivalence. But what can be concluded is that if you want to look at macrophages and the role of steroid hormone imbalance in macrophage infiltration, or you want to model foam cells, you definitely want to work with the ventral prostate. And so obviously a lot of further questions come up from this study. What is the origin of luminal macrophages? So is it monocyte-derived macrophages? So are they like newly infiltrated cells, or just these tissue-resident macrophages decide to go to the lumen? And I put in smooth muscle cells here because foam cells are generally looked at in terms of atherosclerosis. And what they have shown is that more smooth muscle cells are actually able to transform into foam cells than macrophages, and they take up the CD68 marker and everything. So we really have to be careful of like looking at all the different cell types that can make up these luminal foam cells. And we also don't know if foam cells are protective or they drive BPH pathology. So we will find, our approach will be to do single cell RNA-seq and single cell proteomics to identify if there is one subset of foam cells and whether there is a disease related type of foam cells in the prostate in mice and humans. And so it's also very interesting to find out, it would be interesting to find out what drives the translocation of macrophages to the lumen. And this will be modeled by an in vitro microchannel model, whereby you can actually track track macrophages going into the lumen. And also, lipid accumulation is not something that has been really looked at in terms of BPH, especially not the regionality of lipid accumulation in BPH tissues. So we are going to conduct spatial distribution studies, and we will look at modulation of lipid synthesis pathways, as we saw from our experiments that at least one gene is upregulated in this model, but it would be good to map out the changes in gene expression patterns for lipid synthesis pathways. And also, we are going to look into different animal models of comorbid factors that can be really important in further researching BPH. And one is atherosclerosis. So foam cells are really important. They have been discovered in atherosclerosis. And there has not been any studies done in terms of correlating atherosclerosis with BPH. And that's going to be one of the focus of our future studies to identify whether there is a connection. And the other connection that we want to further decipher is the connection of sleep fragmentation with disease development, with dysfunction development, because there are environmental factors, that you work hours that you can't sleep enough. But the other thing is nocturia is something that already happens with people with LOTS. And so is that something that will actually cause more inflammation and will cause progression of the disease? So I would like to thank all my collaborators and my previous mentors and current mentors. And I really appreciate the funding from NIDDK. And thank you.
Next speaker will be Dr. Doug Strand, who is from UT Southwestern.
All right. Good afternoon, everybody. Thank you to the organizers for inviting me. It's a pleasure to be here, see everybody in person. So Dr. Starr talked a little bit earlier about begging, borrowing, and stealing. I'm the guy you want to do that from. I'm going to tell you exactly how you can get your hands on these tissue and data resources for human lower urinary tract research, and I'll leave the basic science of our O'Brien Center to Dr. Macoska and Dr. Vezina afterwards. All right, so my laboratory is interested in the anatomy and cellular pathogenesis of lower urinary tract dysfunction. And that includes the bladder, as shown here in this sagittal section of a male lower urinary tract, which we've largely ignored, but we're now starting to build some data sets that perhaps can give us some insights into the secondary effects of obstruction on the bladder. On the left-hand side, you can see a nano-CT of a human prostate that shows you the ductal network of a human prostate, a normal human prostate. We're also doing this on BPH specimens as well, so we can understand the anatomical structure of benign prostatic hyperplasia. So we know that this is an age-related and progressive disease. You can see the images of the gross images of a normal and hyperplastic prostate in the middle here. So you can see this is a 49-year-old man who has BPH, but probably doesn't have any symptoms yet and may not even know that he has BPH. And this is the earliest form of the disease, where you can see a little bit of hyperplasia around the urethra. The lower urinary tract symptoms, you can see by the green line, trail the histological presentation of BPH. Now, we have very few options for these men. Mostly these guys are treated with alpha blockers just to relax the entire lower urinary tract, help with voiding. Some of these men are treated with 5-alpha reductase inhibitors if the prostate volume is over a certain size. That also provides some level of relief, but isn't targeting the actual disease etiology. So one of our first questions was, are there different cells in this transition zone versus the peripheral zone? And before I get to how we answered that question, I wanted to talk a little bit about how we're actually defining cell types. So I'm a member of HubMap and GoodMap and the Human Cell Atlas. And what we're trying to do is connect anatomy to cell types. So as we discover new cell types through single cell RNA sequencing, we're connecting it to anatomical structures as well. So UBRAN is the consortium that has been creating anatomical definitions for a long time. And more recently, the Cell Ontology is trying to incorporate its understanding of cell type into anatomy. So here we have an example of this from a Nature Cell Biology paper last year, where you can see a hepatic sinusoid that has endothelial cell, which is defined, which is a specific cell type defined in the Cell Ontology. A lot of the new data that's being produced by single cell RNA sequencing has not actually been incorporated into the Cell Ontology. And so that is my role as the prostate and bladder expert, is to help define these functional tissue units and the specific cell types that are in each of these. So based on an Opportunity Award back in 2017 through the University of Wisconsin O'Brien Center, we learned how to do single cell RNA sequencing. And so we set up a collaboration with the local Transplant Alliance, and long story short, we were getting fresh tissues from typically young traffic accident victims, and we performed single cell RNA sequencing on different anatomical parts, the transition versus peripheral zone, and discovered a couple new cell types that were in the prostatic urethra, which is shown by keratin 13 staining. You can see the prostatic urethra lit up in white there. And so now we have an actual cellular definition of the transition zone, which is that transition of prostatic urethral cell types, the new club and hillock cell types that we discovered, into prostate specific epithelial cell types in these proximal ducts. So just to give you an overall hierarchical definition of human prostate cell types, you can see there's an UBRAN term for prostate and an UBRAN term for stroma and epithelia and prostatic urethra, but a lot of these cell ontology terms are actually in process based on the new data that we're creating. And so we have 3 different types of smooth muscle, one of which, the prostate smooth muscle, is in the Cell Ontology, but vascular smooth muscle is not in the Cell Ontology. So this is an ongoing process based on the single cell data. And here is an illustration from HubMap of a prostate functional tissue unit, they're calling, with all of the new cell types incorporated into it. Okay, so in addition to this, so all of those data, I wanted to mention that all of the single cell data and all of the raw images for all of the anchor genes that are identifying each of these cell types in situ are found on the GoodMap database. That database is going to be shut down, and there's going to be a new Atlas Center database where all these data will be transferred. In addition to that, we are creating a human bladder reference atlas, which we are nearing completion, but is currently unpublished. Again, from these young organ donors, we are doing single cell RNA sequencing, validating these clusters that we're discovering as unique cell types or not. This is a process, and there has yet to be consensus around the naming of a lot of these cell types. So that's something that's ongoing. We've also started to do single nuclear sequencing, which allows you to do RNA and ATAC sequencing on frozen specimens, which is a lot easier than doing single cell RNA sequencing on fresh tissue. So given our data, our tissue repository, we're able to go back and now produce single nuclear data from all of these specimens across the aging spectrum, better representing diverse ethnicities, and we can incorporate age-matched matched specimens to our disease specimens that we're getting from our urologists. We're also creating a browsable database of these clinically annotated specimens. It's a web interface called Open Specimen. A lot of universities are using this now. We're inventorying everything we have now, and that will be accessible for everybody to see without having to email me. And then I'll let Chad talk about this in his next talk, but as part of a new RC2 that we're working on, where we're making these data available, he's also going to create new lineage tracer models. He'll talk to you a little bit about the existing ones that he's using to study fibrosis. So with that, I'll say thank you, and maybe we save a little time. These are the people.
Thank you, Dr. Strand.
Okay. Up next, we have Dr. Chad Vezina, and he's at UW-Madison.
So after that talk, it's no surprise that Doug has a very large brain. So several years ago, I begged Doug if we could borrow, steal, kind of the same thing, right? Some of his brain power and collaborate on a project that we were working on with fibrosis. And it's been a great collaboration. Doug has the more difficult part of that, which is the discovery. He uses omic-based approaches to identify potential factors that contribute to BPH, LUTS, and lower urinary tract dysfunction. He drives associations between those factors, and then he hands it over to our group to do what oftentimes is the easy work, which is to determine mechanisms. We use models that we validate together to make sure that they're meaningful for humans, and then we do validation at the end using human samples. So this is just a example of one of the projects that we're working on where we're using that type of an approach. These are transverse sections through human prostates. They have been stained with picrocereus red, which is a dye that has high affinity for collagen, and then visualized under fluorescent light. So everything that you see here in red represents collagen. The top image is from a male that was an organ donor in his 20s. And the bottom image was obtained by, it was a prostate obtained by simple prostatectomy from a man in the 70s who was symptomatic. And you can see that there's pretty substantial differences. I want you to look at the bottom, and I think the arrows on this thing don't work, but if you look on the right side, you see sort of a large sphere. And Jonathan introduced the idea of epithelial nodules contributing to benign hyperplasia and prostate enlargement with age. And that sphere, which is mostly dark in the middle, represents an epithelial nodule of BPH. And you can see that actually it has very little collagen. And so what many investigators confuse about the work that we're doing when we're studying prostate fibrosis as part of the UW-Madison O'Brien Center is that we're not studying collagen and fibrosis as it contributes to enlargement of the prostate. We think that accumulation of collagen is in and of itself a risk factor for developing voiding symptoms and voiding dysfunction, and probably a separate disease. And these are arising potentially together in the same prostates of the same men. Several years ago, I had a graduate student, Kyle Wagner, who was quantifying collagen content, and we looked right in the middle of these specimens and worked our way out along a ruler and quantified what he called the picrocereus red brightness, which is the intensity of stain as a surrogate for collagen density. And what you can see in the donor off on the right-hand graph is that pretty quickly, as you move away from the urethral lumen, the collagen density goes down. But in the man who had LUT-D, the prostate collagen content remains remarkably the same across 5 millimeters of tissue, and it tends to stay high beyond that. A lot of the work that we're doing that's based on fibrosis is based on seminal work by Joe Macoska, who has shown that the stiffness of the prostate and the prostatic collagen content correlate or associate with lower urinary tract symptoms in men. We know that this is above a correlative relationship. This is unpublished work shared with me by Teresa Liu and Will Rickey. And this is looking at a mouse that carried a mutation in collagen 1A1, a predominant form of a fibrillar collagen. And this mutation rendered the collagen protein resistant to proteolytic breakdown. So over the course of life, that collagen accumulates over time. It's never being broken down. And that accumulation was associated with urinary retention. In the right-hand graph, you can see the bladder is being pointed to by a black arrow, and it's much larger. It's filled with urine compared to the left-hand graph. So a collagen accumulation by itself is sufficient to cause voiding dysfunction in these mice. We've been working with a mouse model whereby we instill uropathogenic E. coli into the distal tip of the urethra. We obtained this E. coli after seeing a presentation from Jonathan Barash in this group several years ago, but Indira Mizo-Rikar was initially one who made this strain of E. coli in collaboration with Scott Hultgren. And when we introduce this into the distal tip of the urethra, it ascends into the prostate. It infects the prostate. It drives inflammation. It's heterogeneous inflammation. So we see pockets that appear to be histologically normal, some that appear to have inflammation, and the areas that have high inflammation also have the highest level of collagen. Collagen density is significantly increased. We know that collagen is increasing, not because collagen is accumulating and not being broken down, but instead because collagen is being newly synthesized. And we know that based on RNA analysis. This is work by Brandon Sharp, which shows collagen 1A1 mRNA abundance increases in this model of prostate inflammation. So this gives us an opportunity, now that we have a model that recapitulates at least some aspects of the histological inflammation and collagen accumulation that we've seen in men, it allows us to answer a question. The question we wanted to answer is, what is the cell type that's giving rise to the collagen in the prostate? If we can understand what that cell type is and what drives that cell type to produce collagen, we can reveal multiple pathways by which we can intervene pharmacologically. And so we used an approach called lineage labeling. And for those of you that don't work with mouse models or genetics, this involves two genes that we smash together into the genome of a single mouse. The first gene is a bacterial gene called KURe recombinase, and it recognizes short DNA sequences, two short DNA sequences, and it cleaves out everything that's between them and it sews them back together. The other allele that we introduced into these mice is a red fluorescent protein reporter gene, and it contains a stop codon right at the start of the gene. And the stop codon is flanked by these sequences recognized by KURe recombinase. So if KURe recombinase is expressed in the cell and this reporter gene is expressed in the cell, the KURe recombinase will chew out the stop codon and allow the RFP to be expressed. It's not only expressed in this cell now, but if the cell differentiates, it persists in the cell. If the cell divides, it is sent down to the offsprings of those cells. So we can follow that lineage through a disease process to determine what are the cells that are giving rise to collagen. We begged, borrowed, and stealed again. We asked, because we were sort of behind the game when thinking about fibrosis in the lower urinary tract, we asked about other organ systems that have been studied more readily with respect to fibrosis, and what are the cell types that have been identified there that might contribute to fibrosis. And that allowed us to hone in on 6 different KURe expressing mouse lines that would hit on these different cell types. Some of them are relatively specific. The steroid 5-alpha reductase type 2 KURe shown at the top is specific for prostate fibroblast. There are others like this GLE1 KURe, which is expressed in perivascular cells known as pericytes, but it's also expressed in bone marrow derived cells. It's one of the limitations that we have in doing lineage tracing right now, is that the KURe lines that we have are not necessarily, though they're selective, they're not specific for single cells. And that's something that Doug and I are working on as part of an RC2 grant going into the future to give more specific KURe expressing strains for prostate and bladder. So just to give you an idea of outcome of this, this is after we've smashed together these 2 alleles and then introduced E. coli and taken out the prostate and sectioned it and stained it. We stained it for collagen, which is shown here in white, and for the red fluorescent protein, which is shown in red. And these are 2 different examples. On top, that's a lineage that we traced that was supposed to be specific for smooth muscle. You can see no overlap between the red and white. So those red cells, those smooth muscle cells, don't contribute to collagen synthesis in the inflamed mouse prostate. But the bottom lineage, this is a LYZ2 lineage, which is known to be expressed in myeloid cells that originate from the bone marrow. You can see that all of those red fluorescent protein positive cells also co-express collagen. So looking at this as a function of total cells, We had several that contribute substantially to the overall production of collagen. There's a GLI1 CRE that contributes. There's a S100A4, which typically marks fibroblasts, but also bone marrow cells, and the Lyse2, as I just mentioned. CD2 is a marker of lymphoid cells, and that contributed a small amount. Now, what was confusing to us is that we would predict that, maybe there are multiple cell lineages that contribute to the overall collagen synthesis. And when we add all of those together, they'll equal 100%. But even though I'm from Wisconsin and my math is a little questionable, you can see that all of those add up to far more than 100%. And so what that suggested to us is that these are probably not marking unique cell lineages, but instead they might represent the same lineages. These genes could be co-expressed in the same cells. And I just want to mention that Jaskiran Sandhu and Sneha Chandrasekhar are going to be presenting a poster in the next session, the first session tonight, poster number 9. Okay, so save your hard questions for them. So we used a method where we can label RNA in these lineage labeled cells, and we did this across multiple lineages. And what we consistently were able to observe is that cells that were expressing RFP driven by Lyse2, for example, those also harbored GLI1 mRNA and S100A4 mRNA. And cells that were expressing GLI1, they also harbored Lyse2 mRNA and S100A4 mRNA. So these are really the same cell types that we're talking about that are producing collagen in the prostate. Now, we have used a lot of Doug's Strand's approach of using single cell RNA sequencing to identify these collagen producing cell types. But there are some limitations with single cell sequencing. You have to digest the prostate into single cells. They need to survive, they need to be healthy, and only those healthy cells are sequenced. Sometimes cells are susceptible to the proteolytic and mechanical digestion that happens and they die, and you never get a chance to sequence them. And so while we weren't seeing cells that co-express Lyse2, S100A4, and GLI1 mRNA in Doug's single cell RNA sequencing profiles, when we mapped them back onto sections where we hadn't manipulated the tissues, we simply fixed them, we were able to show that there are cells in the human prostate that co-express Lyse2 and a version of collagen that's expressed within the cell. So we think that this cell type that we identified in mouse is likely also present and an increase in abundance in inflammation and producing collagen. Okay, so then we wanted to ask the question, if these things are coming out of bone marrow and circulation, how are they getting to the prostate? Because that might be a druggable target as well. And so this is some of the work by Brandon Scharf. He went in and did some RT-PCR, and he determined that a specific chemokine called CCL2 is increased in abundance. Oh, this is CCR2, but CCL2 is also increased in abundance in response to E. coli in the mice. Unfortunately, we decided to surround ourselves with a bunch of smart people that have nothing to do with prostate research, and we meet with them once a week. We meet with a bovine lactation biologist. We meet with somebody who studies craniofacial morphogenesis and cleft lip and cleft palate. We meet with somebody who studies breast cancer, and it's a great place to exchange ideas, to practice explaining your work in front of a diverse audience, but also to learn new techniques that you might be able to steal and apply to your own work. And so in this particular work, we listened to a talk by Lisa Arndt, who's a mammary gland cancer biologist. And she was studying how obesity increased the risk of breast cancer, specifically by causing an increase in fibrotic tissue. And she had some resources that were very interesting to us, including a mouse that would, under some conditions, express RFP driven by the promoter for the CCL2 receptor called CCR2. Or if we manipulated the genetics further, we could create a null allele so that they didn't have a functional CCR2. So the important thing to see here is that RFP marks these CCR2 positive cells, and you can see they greatly increase in number in the E. coli inflamed prostate compared to the control prostate, and they co-express collagen. We decided to knock out CCR2 or deliver 2 null alleles to the genome. And under those conditions, we were surprised that we didn't get rid of inflammation altogether. We still saw inflammation. We saw an accumulation of CD45 cells, which is a general marker for leukocytes, but we reduced the number of RFP and white positive cells, which reflect collagen synthesis. And we then decided we would analyze collagen fiber density in these mice. And so just to show a couple of these data first, the far left there is under conditions of no E. coli and no inflammation. The middle is a wild type control mouse where we delivered E. coli to cause inflammation. And the far right is a mouse where CCR2 function has been ablated and has been infected with E. coli. So while they still are prone to inflammation, these CCR2 null mice do not accumulate collagen. We thought that was very interesting. And if it truly is a circulating cell that's giving rise to collagen synthesis, we should be able to add it back to the mouse. So Hannah Ritten cracked the femur of some wild type mice. We sucked out the bone marrow, we purified it, we injected it into circulation of these CCR2 null mice, and then gave them an E. coli infection. And under conditions where there was no inflammation, we saw about the same amount of collagen fibers. But when we had knocked out CCR2 function, we then restored collagen, the fibrotic response to E. coli. So it truly is a circulating cell, and CCR2 has something to do with this story. I wanted to highlight Brandon Sharp, who's likely to be now changing sessions. He was poster number 41, but he's likely to be changing sessions, and he's going to tell you more about his story in tracking down the mechanisms that recruit these cells and factors that induce them to produce collagen. So the bottom line there is that we've identified a cell type. It's a bone marrow derived myeloid cell that is recruited upon E. coli-induced inflammation into the mouse prostate that appears to increase in abundance and high-grade inflammation in the human prostate and is responsible for collagen synthesis. And I just wanted to thank all of the people in our lab that are involved. They're all here today. Fresh new picture. Airport is like one of the first times that we actually got together, but everyone's here and working on different projects. So please go see their posters. Thank you.
All right, our next speaker up is Dr. Joe Macoska, who is from UMass Boston.
Okay, running right on time here. I'll try not to mess that up. So let's talk about fibroblast plasticity in the lower urinary tract. First of all, what is a fibroblast? You've been hearing about fibroblasts basically all afternoon, but they're broadly defined as connective tissue resident cells that produce the extracellular matrix scaffolding of the body. However, they're very heterogeneous, and I think we have to keep this in mind as we try to make sense of, basically all the findings that you're hearing today. There is no pointer here, but I think you'll just have to look at this. They differ in terms of what developmental lineages led to their production in the first place. They're different in terms of their molecular phenotypes. They're different in terms of how they respond to their microenvironmental niches. And all of this can influence fibroblast plasticity. And in fact, the latter one, how they respond to their microenvironments, can also alter their molecular phenotype. So they're not written in stone. They don't come out one way and stay that way. They can change up. And I think that's where we get some pathobiology. So what can modulate the prostate microenvironment? There's quite a few things, but you've heard about some of them already today. One thing for sure is diabetes or metabolic syndrome. Dr. McVary talked about that, and I think that's very appropriate because if you look at epidemiological studies, this is one of about 5,000 men, you see that men who reported or actually were clinically diagnosed with metabolic syndrome were disproportionately seeking treatment for LUTS very significantly compared to men who did not have metabolic syndrome, and they had more severe lower urinary tract symptoms. So there's a clear association here. We see also that if we kind of throw into the mix the idea of aging, we can come up with something that's being called inflammaging. And this is actually a combination of what happens when you have aging, metabolic syndrome, and diabetes all happening at the same time. And the answer is nothing very good for the person who is experiencing these. And so here you see a picture of an individual who has what we call visceral fat accumulation or abdominal fat. You can think of it that way. But this is actually where we see metabolic syndrome being manifest and also an increased risk for type 2 diabetes. And often it is consequent to aging, or is in association with or concurrent with aging. Let's put it that way. So when all of this is happening, the adipocytes in the fat and all of the infiltrating immune cells that are drawn to that begin to make kind of a gemisch of pro-inflammatory adipokines and cytokines. And this is really the manifestation of inflammaging. And the downstream results of that are all of these pathobiologies you see coming off of that lower arrow. And I think there's a lot of data now suggesting that fibrosis is actually one of these things that can result from inflammaging. And so what does inflammaging else actually do? to the microenvironment that might induce this fibroblast plasticity, and that will lead to things like fibrosis. One thing that inflammaging can do is promote what's called cellular senescence. You may have come across this at some point in your studies, but basically senescent cells are ones that leave the cell cycle. So they're not replicating anymore, but they're still metabolically very active. And they can occur as a consequence of some kind of DNA damage processes, replicative exhaustion, their telomeres get real short, or stress. And with inflammaging being bombarded by all of these cytokines and inflammatory mediators causes stress on the cells in the microenvironment, including fibroblasts. So they may undergo senescence, and when they do, they exhibit what's called the senescence-associated secretory phenotype, or SASP, where they begin to actually secrete all of these cytokines and inflammatory mediators that kind of push them into senescence in the first place. And so they actually start to take on a different phenotype because this is not how they normally are. And when that happens, they start to change their microenvironment. So now they're not just the victim, now they're the agent. And cells around them may start to undergo senescence. And together they're going to be secreting these inflammatory mediators and cytokines that are going to become part of the microenvironment, and they were not there before. And you can see what happens here when that process occurs. This is a very nice study, one of many, by the Judith Campisi lab, where she actually forced different cell types, fibroblasts and epithelial cells in the prostate to undergo senescence, and looked at how that changed their transcriptomes and secretomes. And if you probably can't read this from where you are, but when these cells become senescent, you can actually identify the exact kinds of inflammatory mediators and cytokines that they begin to express that they were not expressing before. And actually, they're now secreting things that you usually associate with immune cells, but these are fibroblasts. And so they're kind of taking on a bit of a different phenotype, and in fact, their molecular phenotype is changing consequent to senescence. And so we saw this in my lab as well. This was a while ago now, but we wanted to know if fibroblasts from older men that we define as older than 60, sorry if any of you are in that group, we just had to, make a cutoff, and those who are younger, which is younger than 60, if they were actually expressing the same kind of transcripts. And we found out that they were not. And in fact, the fibroblasts cultured from the prostates of older men, and this is the periurethral area, were actually expressing all of these CXC type chemokines that you would not expect them to be expressing at all. That's really more of an immune cell transcriptome. And in particular, they were expressing CXCL12, which is stromal cell derived factor 1. So we started to look at what are the consequences of expressing so much CXCL12 in that environment. And we identified that in fact, CXCL12, when it binds to its receptor, can transactivate EGFR, and through different signaling pathways, mainly MET-ERK and PI3 kinase, will turn on the collagen 1A1 and 1A2 genes from which the collagen protein will arise, and alpha smooth muscle actin. So we were getting extracellular matrix deposition, and actually those fibroblasts that were being exposed to this were now actually phenoconverting to myofibroblasts. And this is a different pathway than the one you associate with TGF-beta, but the end result was the same. Okay, these fibroblasts were getting activated and started to secrete lots of extracellular matrix. So they were responding to their microenvironment in this way that's consistent with fibrosis. We also saw in vivo in the mouse, this is on the left, that if we induced metabolic syndrome in mice by feeding them a high-fat diet, they expressed way more collagen in the periurethral area of their prostates than mice who were fed a low-fat diet. And on the right, again, I wish I had a pointer, you would see that with the high-fat diet, these mice were experiencing urinary voiding dysfunction, much more so than those fed a low-fat diet. If we hit the mice with a CXCR4 antagonist, which what we saw then was that the high-fat diet-fed mice, the collagen accumulation was reduced because the CXCL12-CXCR4 axis couldn't signal, and the urinary voiding function became more normal. And so you can see that actually there's a consequence to things like senescence, which are consequent to metabolic syndrome, inflammation, inflammation, and diabetes. Okay. Recently, we started to look at a different group of those proteins, the inflammatory mediators, that we see consequent to senescence, and these are the interleukins, and specifically IL-4 and IL-13, which we studied together because they signal through the same signaling axis. And we saw something a little different with IL-4 and IL-13 in that they would promote the expression of collagens by human prostate fibroblasts, but not alpha smooth muscle actin. And this meant that they were not differentiating all the way to the myofibroblasts, which you see in the bottom there, but they were stopping before that at a condition or I guess a phenotype, which is called an activated fibroblast. And so they have actually changed their phenotype, but they are activated, they're making extracellular matrix, but they're not fully phenoconverted. And that has some consequences. One is they can still replicate. So they're not terminally differentiated, so they can accumulate. And they can continue to be a source of extracellular matrix production in the prostate. We also saw that they can signal through JAK-STAT signaling pathways. This is the major way that they signal. And again, I wish I had a pointer. I should stop saying that because I don't. But you can see on the right that when we treat the cells with IL-4 or IL-13, but mainly IL-4, it's a stronger one, we see collagen 1 production, we see collagen 3 production, we see extended phosphorylation of STAT6, which is the major transcription factor that's going to turn on the collagen genes. And if we hit that with tofacitinib, which is Xeljanz, okay, this is in clinical use as a JAK-STAT inhibitor, IL-4 can't rescue that production of the collagens or phosphorylation of STAT6. And so this is clearly the pathway that IL-4 and IL-13 are using to make these collagens be expressed. We also saw, and I'm not going to go through all this, but we published this a couple months ago. Quentin Darcy, who's sitting right there, is the first author on this. So if you have questions, you can talk to either one of us. That the signaling pathway not only was responsible for collagen accumulation, but it also produced IL-4 and IL-13. So the initial IL-4 and IL-13 was now being produced by the fibroblasts themselves, extended phosphorylation of STAT6, which is required for that, and expression of the cognate receptors of IL-4 and IL-13. So this is really a very kind of self-contained, kind of like a continually progressive loop that IL-4 and IL-13 set up, which means that this is going to be something that once it gets set up in the prostate would be difficult probably to disrupt. And along those lines, we were thinking if these cells learned how to make IL-4 and IL-13 because they were treated with it, this is something immune cells do. And so we looked at what immune cells do in response to IL-4 and IL-13. And if we're looking at CD4 plus T cells, because they're a rather large population of cells that we see in the prostate, when you treat those with IL-4 or IL-13, they will differentiate into Th2 CD4 plus cells, which also make IL-4 and IL-13 and a lot of other interleukins. And so they also kind of become self-sustaining. And that made us think, are fibroblasts kind of learning how to be immune cells when they're exposed to some of these inflammatory mediators? And if so, then what other things might they be learning how to do that they didn't know how to do before as just naive fibroblasts? This is not a very oddball, well, you might think it's oddball, but it's not that odd of a hypothesis, because the concept of fibroblast plasticity is based on the idea that depending on the microenvironment these fibroblasts find themselves in, they can start to take on the phenotypes of other cell types, including immune cells. And so again, it's very microenvironmentally niche kind of dictated, but it's not something that we should ignore. We should look at it. So one thing IL-4 does in the immune system is it makes immune cells very motile. And actually, when it sets up a gradient, immune cells will gravitate towards the IL-4 gradient. And so we were wondering, does IL-4 induce prostate fibroblast motility? So using scratch assays and kind of a dose response to 40 nanograms per mil or 80 nanograms per mil, of IL-4 over a 16-hour period, we measured the percent of wound closure, which is a measure of motility, and saw, in fact, the graph on the right, I think will show you this quite well, that compared to control, those treated, the fibroblasts treated with an increased dosage of IL-4 did in fact move into that channel, the scratch, and in fact, by 80 nanograms per mil, they were really moving. They were basically closing up the channel. And so they were very much exhibiting fibroblast motility, much like an immune cell. Another kind of concerning thing that IL-4 can do for immune cells is that it can help the cells resist apoptosis, which is a problem. Fibroblast and myofibroblast persistence in fibrosis is a problem that has kind of defied our ability to really treat fibrosis in many cases. And so is IL-4 actually doing this for fibroblasts and making them perhaps resistant to apoptosis? And so we looked at extrinsic pathway apoptosis on the left by looking at caspase-8 activation and caspase-3 activation consequent to treating them with 3 pro-apoptotic proteins, TRAIL, Fast ligand, and TNF-alpha, which we just call TRIO for short. We also did look at intrinsic apoptotic pathway activation by looking at caspase-9 activation. And kind of a long story short, on the right, it's the same data. On the left is the Western blot data, but on the right is ELISA data, which might be a little easier to digest in a short period of time. But if you see vehicle-treated cells, they have a very low apoptotic fraction. They're not going to be dying for any particular reason unless you bombard them with pro-apoptotic proteins. Same when you treat them with IL-4. And so those are very much alive. However, if you treat them with TRIO, you can see that the apoptotic fraction really goes up. And when you actually treat them concurrently with IL-4 and TRIO, you see that the apoptotic fraction is reduced, meaning that IL-4 is protecting these cells from undergoing apoptosis, even in the presence of these very powerful 3 pro-apoptotic proteins. Okay, so I won't summarize all this because we just talked about it, but overall, it does appear that these fibroblasts in the presence of IL-4, and I'm not saying this is the only inflammatory mediator that might be doing this, it's only the only one we've studied like this, is actually kind of modulating the phenotype of these fibroblasts so that they're becoming more like an immune cell. So what are the consequences of all this? So we have some initial immune exposure to IL-4 and IL-13, and that's going to activate the fibroblasts. Okay, they will accumulate and they'll continue to make extracellular matrix and IL-4 and IL-13, and that's going to further activate fibroblasts. Okay, so the surrounding ones can be activated as well, right? These will accumulate and persist because now they're somewhat resistant to apoptosis. Surrounding fibroblasts will see the IL-4 gradient and move in and perhaps become activated themselves, okay? And if this continues, this would then continue to alter the prostate microenvironment and ultimately promote fibrosis. All right, so I want to thank everyone who's worked on this, Quentin, who's here, Marissa Sarna-McCarthy, who's done the apoptotic work. These are both graduate students in the lab, as well as Delaney Bowen, who's been assisting with them, and Alyssa Zillen-Roth, who did some of the IL-4 work that I didn't show you, and who now works at Sanofi Genzyme, which is one of the many organizations that have provided funding for this. So I'll stop there, and thank you.
Thank you. And last here in the session, we will have Dr. Shane Wells from UW-Madison.
Thank you. Thanks for the invitation to speak today. Let's see if I can pull up my talk. Oh, okay. That one.
Yeah, that's it.
Okay, so we're going to move from the micro to the macro with image-based assessment of the prostate. And the title of the talk is Towards Image-Based Biomarkers. I'll back up. I'm an abdominal radiologist at the University of Wisconsin, so I've been clinically heavy for the first decade of my career, and I'm moving into this benign urology space. And it's great to be here. This is my first ever in-person CAIRIBU meeting. The last 2 years I've participated remotely, which has been a great option. These are on the left is an image-based biomarker that's accepted into practice today. We're going to talk more about it. An image in the middle and the right are the biomarkers that we're developing in our at the University of Wisconsin. Right, so these are the objectives. Our goal in our group is to translate, adapt, and develop these image-based biomarkers for the prostate and the lower urinary tract. And I'm going to share work from what I'm doing, and I'm also going to share work from Alejo Roldan, one of my collaborators in engineering and radiology. All right, we're going to start in the liver though. And the reason I think we should start in the liver is because there's many parallels between what I see in the prostate or what I've learned from Will Rickey and the CAIRIBU folks to what I've seen in the liver during my career as we develop biomarkers for non-alcoholic fatty liver disease and NASH. So non-alcoholic fatty liver disease is the abnormal deposition of fat in the liver. And it's on a spectrum from simple steatosis, so that's just fat in the liver that doesn't have any consequences, to steatohepatitis, which can lead to fibrosis and cirrhosis. And these, what you see here, the PDFF stands for proton density fat fraction. This is a map that quantifies fat in the liver. And as you can see on the left, in a normal patient, this patient has 5.5% liver fat, which is normal. Under 6 is normal. And the patient on the right has a very large amount of fat in their liver. As you can see from a morphologic standpoint, you can see that the shape of the liver is even different, right? This liver is enlarged, has a round contour, and the fat content is very high. This is a very common disease worldwide. It's more common in the West, and it's the most common in the USA. Importantly, and what we probably don't discuss enough, is that the susceptibility and progression from NAFLD to NASH is heterogeneous. And its factors include race, ethnicity, lifestyle disparities between these groups. So for example, Hispanic Americans are much more likely to develop non-alcoholic fatty liver disease compared to white Americans. Hispanic Americans compared to Hispanic Americans compared to European Hispanic folks, their progression from their risk for NAFLD is lower. And their progression to NASH is lower. So folks from Europe, Hispanics from Europe don't progress to fatty liver and fatty liver disease as often as Hispanic Americans. It's important to understand those demographic differences. Historically, the diagnosis of NASH was made by biopsy, but you can see how this is a problem, right? It's a very small sample size. It's painful. There's risks, including death, and it's not suitable for screening and surveillance. So the development of NAFLD and NASH and the progression of medications that can help reverse these conditions drove the development of biomarkers in the liver. Quantitative or qualitative imaging, this has been around since imaging was developed, right? This is a radiologist looking at pictures and describing what they see from a qualitative standpoint, right? So the image on your left is a patient with cirrhosis, right? So NASH-associated cirrhosis, the echogenicity of the liver increases, right? I know this liver is echogenic because I've seen thousands and thousands and thousands of livers with ultrasound. But the problem is, if I would grade the degree of steatosis or the degree of cirrhosis, it would be subjective, right? Based upon my personal experience. And there's a lot of inter-observer variability between my assessment and a colleague's assessment. The images in the middle and the right, these are of a patient with a normal liver on MRI and then NASH cirrhosis. Now you can see that there are differences in the way the liver looks on these two pictures, right? But sometimes the differences are much more subtle than this. Oftentimes what we rely on is the characterization of these secondary features, right? So you can see on the image on the right, the patient's spleen is large, right? So that suggests to us that they have portal hypertension. Other things you can look for are ascites, so accumulation of fluid in the belly, or dilation of the portal vein, clot in the portal vein, because as the liver becomes cirrhotic, it becomes stiff and portal venous flow slows down, and that increases the risk for thrombus in the portal vein. Quantitative imaging, though, this is the hallmark for our society as we move forward. And so what is that? That's and the way we define it as a society is the extraction of quantifiable features from images for the assessment of normal and the presence of disease and the response to treatment. So you've seen these images before, right? I'm going to show them to you again because this is one of our image-based biomarkers that we've developed in the liver that is used around the country and across the world. Again, patient on the left has a normal fat fraction below 6%. Patient on the right has a fatty liver. Again, you'll notice those morphologic features, a big liver, and you can tell the color of the liver is different. It's slightly more gray, but importantly, the fat content is very elevated. All right, so elastography. This is the next step of quantitative imaging in the liver that we're currently using. And so what is elastography? Essentially, this is an image-based form of palpation that is quantifiable, right? Reproducible. And so elastography, what we do is we transmit low frequency waves into the tissue, and then we measure them. This is an example of how we do it with MRI, but we can also do it with ultrasound. And again, this is the patient you've seen before. They have a normal looking liver on qualitatively on MRI. You can see the wave image in the middle and then the elastogram, which is the stiffness map. You can see this patient has a liver stiffness of about 1.8 kilopascals. Below 2.5 is normal. So when the liver is purple, as you can see here, that's generally normal. All right, so in NASH, inflammation leads to immune cell infiltrates, myofibroblasts, and collagen formation, similar to kind of some of the stuff we've already heard today about what happens in the prostate in the presence of metabolic risk factors. Now, this is the elastogram of the other patient you saw that has elevated liver fat, right? They have a large liver, and the fat content is elevated. And as you can see, the stiffness map in the middle, the color is not purple anymore. It's blue, green, and yellow. And the stiffness is increased to 2.9 kilopascals, which is in the inflammatory, in the inflammatory / stage 1 category. And I put normal there beside the image just to kind of show you the visual difference from a qualitative standpoint. But importantly, we can measure this stiffness and follow it over time. NASH can lead to fibrosis and cirrhosis, and ultimately to decompensated liver disease and mortality. This is what NASH-related cirrhosis looks like as the patient progresses through the stages of liver fibrosis and ultimately to cirrhosis stage 4, right? So the liver goes from 3.2 kilopascals at stage 2 to 4.8 at stage 3, and then a very stiff liver, 7.9 kilopascals, right at the top of the spectrum at the stage 4 fibrosis, the end stage disease. Now, MRI is expensive and it's not universally available. So ultrasound is often used more often in surveillance, or commonly in surveillance. And this is an alternate way to also determine liver stiffness. You can see that the big difference is with MRI, you get a global look at the liver. You see the entire liver, and you can see, if I go back a slide, you can see the marked heterogeneity and stiffness in the right liver compared to the left liver. Now, that's not possible with ultrasound. You get more of a point assessment, so similar to a biopsy, but it is useful for surveillance because it's much cheaper, and it's almost universally available across the country. Right. In this patient, the images in the middle and the right are shear wave images. Those are estimates of, they create estimates of liver stiffness. And you can see at baseline and 1 year follow-up, the liver went from moderately stiff to more normal with lifestyle modifications in a patient with NAFLD and NASH. All right, so let's get to the topic at hand, which is BPH and LUTs. Now, this is stuff you all know in the room, and you probably know better than I do. But importantly, imaging has had a historically limited role in the assessment of BPH LUTs, right? This is what's commonly done in clinical practice. You'll get an ultrasound to check post-void residual in the bladder, and you can do a transrectal ultrasound to kind of get a size estimate of the prostate, which we know now through work with Dr. Grimes, who's in the room, that that is a poor assessment of prostate volume. There are better ways to do it. Now, aging, as you know, and inflammation can contribute to increase in prostate size and cellular changes. And metabolic risk factors are important. So qualitatively, when we look at prostate MRI, similar to liver MRI, qualitatively, as the liver develops fat and then in NASH and then cirrhosis, the morphology of the liver changes. Similarly, the morphology of the prostate changes from normal. And I'm going to go back a slide so you can see what normal looks like. So normal prostate there on the left, you can see the peripheral zone is a light gray color, and it's thick, and the transition zone is very small. In fact, it's difficult to distinguish in this normal patient. Image on the right is a patient with BPH, right? So most of what you see there is the transition zone. The peripheral zone is very thin. It's effaced. And there, in this patient, there's both epithelial and stromal heterogeneity within the transition zone, right? But patient to patient, on MRI, you can see heterogeneity in patients who have BPH and symptomatic LUTs, right? These patients each have their prostate volumes are different, right? And the appearance of their transition zone is different. The patient on the left has much more stromal proliferation compared to the patient on the right, which I showed you previously. Oh, importantly, as the prostate grows, the thickness of the fibrotic capsule increases, and right. So as the prostate grows, the capsule thickness prevents further growth, and that increases pressure in the prostate. So that's why we think one reason why determining stiffness of the prostate is important, in addition to the heterogeneity, stromal and epithelial proliferation, and collagen deposition. We think that as the prostate grows, that's another important factor that increases pressure and then alters urodynamics. So this is an elastogram. You saw this at the opening slide. This is what normal looks like. Patient on the image on the left is just the T2 weighted MRI. You saw that image before. Patient image on the right is the elastogram. And what I've done there with the purple is outline the prostate, and with the black, I've outlined the transition zone, right? So that you can see here that we can differentiate the zonal anatomy of the prostate with elastography. And if I measure those, the peripheral zone stiffness is about 3 kilopascals, and the transition zone stiffness is much higher, about 7. Now, this is a normal patient, right? So, this is, I'm going to show some work from Alejo Roldan, one of my collaborators, who is a former Opportunity Pool Awardee and now holds our R01 from the NIDDK to do this investigation that I'm going to show you now. This is a dynamic MRI of the bladder and urethra during voiding in a normal subject. So the patient I showed you before was normal. This patient having a urodynamics with MRI is also normal. Notice that as the bladder empties, the urine is the white color on the image on the left. You can see the bladder empties rapidly, relatively uniformly, and there's very little post-void residual, very little of the white stuff left on the image. And you can see that this process of monitoring the urination dynamically with MRI follows very closely with a uroflow right? Now, this is what the voiding a flow rate in the urethra looks like graphically in a normal subject, right? So you get relatively rapid acceleration in flow rate, a plateau, and then a rapid deceleration during the voiding process in this patient, which lasted about 90 seconds. Now, if you superimpose the urethra, this is the prostatic urethra. If you superimpose that on the image, you can see visually differences in the flow rate. And what I want to point out is the relatively capacious blue part is the prostatic urethra. And just below that, the more narrow red segment, that's the membranous urethra. So in this normal patient, flow rates are the fastest in the most narrow part of the urethra, which is the membranous urethra. But let me move to an abnormal patient so you can see what that looks like at imaging, both from a stiffness standpoint and then from a bladder biomechanics standpoint. Now, you've seen this patient before. You can see that their prostate's enlarged. They both have stromal and epithelial hyperplasia. The image on the right is it's a type of shear wave elastography called ARFE, but it also estimates stiffness. And on this image, bright equals soft, dark equals stiff, right? So And most of what you see here on this coronal image is the transition zone. The peripheral zone is very small in this patient and hard to see. But what you can see is there are a bunch of nodule formations here, presumably epithelial nodules, right? They're soft like the peripheral zone. We don't see many stromal nodules in this patient, which would be darker. And number two, you can see around the expected location of the urethra, there's very dark signal, right? Perhaps that's stromal or stromal proliferation and or collagen deposition. And that can affect urethral opening, right? So if we go to this, this lower urinary tract biomechanics again from Alejo's group, you can see that this patient with BPH and LUTS, voiding looks very different, right? The bladder empties more slowly. It doesn't completely empty. There's a very large post-void residual. And the flow map looks different, right? There's rapid acceleration of flow during voiding in the urethra, but a relatively slow drop-off as the pressure in the bladder falls and flow rate decreases. And we think that's because the urethra in patients with prostanomegaly and other factors just doesn't open as well. Now, this is a superimposed, this is the urethra showing visually the flow rate through from the prostatic to the membranous and the penile urethra. And you can notice that there's the red, so the prostatic urethra is more narrow and the flow rate is higher as it extends through the membranous urethra, right? So the urethra doesn't open as well, and that results in a kind of like a stricture, accelerated flow rates, and we think that it impacts the bladder emptying, right? So just one other thing that we're working on right now, we just started working on is understanding better what BMI is. We think BMI is a poor classifier of obesity, and what we want to do is to dig into the specifics of what causes metabolic risk factors. And we know those are sex, and men are more subject to metabolic risk factors than women, race and ethnicity, which we've discussed before. But there's other quantifiable risk factors like fat in the liver, myopenia, muscle size, muscle density, those factors. So we're using automated techniques to quantify visceral and subcutaneous fat, muscle mass, muscle density, and then MRI to quantify liver fat in an attempt to create nomograms where we can better understand risk factors of metabolic risk factors for patients who may progress to have lower urinary tract disease. So we think these are important things, and we've had a lot of fun working on them. And thank you for your attention and time. I'd like to really thank my collaborators here, Will Rickey and the O'Brien Center for all the support. And the Opportunity Pool Fund, the Opportunity Pool Award that I was given to do this research. And Alejo just texted me and let me know we're doing a small clinical trial, and our first patient signed up next week. So we're going to image our first patient next week in a clinical trial, the elastography and urodynamics clinical trial. So thank you.